I'm Nicole. Today I'll be sharing about our approach to cancer drug response prediction in patients via semi-supervised deep co-training. Precision oncology aims to find the right drug for each cancer patient based on their unique genomic profile. This is done by predicting patient response to various drugs. However, this is hindered by the limited number of labeled patient samples available for training drug response prediction models. Prior methods have relied on cell lines cultured in controlled laboratory environments, where the response to various drugs can be experimentally measured. These cell lines, however, differ from patients in terms of the distribution of mutations observed and in the way the response is measured. The real-valued AAC response measured in cell lines have no good correspondence to the RECIST categories measured in patients. Transfer learning approaches have focused on handling these differences across the source cell line domain and the target patient domain. Prior literature on transfer learning can be divided into three: inductive, transductive, and unsupervised, based on the use of labeled and unlabeled source and target domain samples. Unlabeled patient data has been mostly used in pre-training models and less in the downstream drug response prediction tasks. In addition, most of the prior methods have focused on using transcriptomic data, which is often unavailable in clinics. This makes the direct translation of prior methods to a clinical setting difficult. In this work, our research objective is to predict cancer patient response on drugs using only data available in FDA-approved clinical sequencing panels. This includes mutations in specific genes and copy number variations. We aim to develop clinically translatable models. To do so, the key considerations include accounting for biological discrepancies across cell lines and patients, as well as utilizing unlabeled patient data. Decoder is a semi-supervised domain adaptation or SSDA technique that considers these. SSDA aims to adapt the model to the target domain given only a small set of labeled data from the target domain and a large set of labeled data from the source domain. Although SSDA allows for two sources of supervision, the source and target domains, the challenge lies in the fact that the amount of labeled data from the target domain is much lesser than that from the source domain. Furthermore, the domains inherently differ in their distributions. As such, a single model trying to learn from both domains can be easily dominated by the labeled source data and fail to effectively make use of the labeled target data available. Decoder learns two distinct models for two tasks: an unsupervised domain adaptation UDA task and a semi-supervised learning SSL task. UDA and SSL tasks are correlated and complementary tasks, which can learn from labeled source and unlabeled target domain data. Decoder also employs pseudo-labeling in its algorithm to train a separate model in each of the UDA and SSL tasks. Each model predicts on the unlabeled data from the target domain to use as pseudo-labels. Since the two models learn from different sources of supervision in their task, they output different predictions with different levels of confidence given the same data. Given a threshold for pseudo-label selection set as a hyperparameter, each model's prediction that has confidence above this threshold is added to the training set of the other model to retrain on. Hence, the strengths of the two models are integrated by having their highly confident predictions exchanged with one another to teach each other iteratively. From the perspective of a teacher-student architecture, each model is simultaneously playing the role of both a student and a teacher. Specifically, each model learns from the pseudo-labels of the other model and also provides pseudo-labels to the other model to train on. By having one model to give confident pseudo-labels to the data that the other model is uncertain about, the strengths of each model can be integrated to jointly learn stronger models that perform well in the target domain. Since we decompose the two sources of supervision available from the two domains, cell lines and patients, into two distinct subtasks, this provides us with the benefit of preserving the nature of cancer drug response prediction problem in each domain. Learning a separate model for each task allows us to retain the nature of the regression problem for cell lines, as well as the binary classification problem for patients. The UDA task is a regression problem where the model predicts cancer drug response as continuous AAC values. On the other hand, the SSL task is a binary classification problem where the model predicts cancer drug response as binary values. So what are our experimental settings? We experiment with the inputs to the model for each drug using mutation, CMV, and a combination of mutations and CMV available in FDA-approved panels. We use cell line data from Cancer Cell Line Encyclopedia, CCLE, and pan-cancer patient dataset from the Cancer Genome Atlas, TCGA. All our experiments were evaluated using AURC on patient test set. To compare performance, we also trained a neural network only on patient data to use as a baseline, shown here in green, and these are our results. What can we infer from our results? We see that on average, using both mutations and CMV yields better results than using either one individually. When only CMV is used as input, Dakota outperforms the baseline and velodrome. When CMV is included, the baseline outperforms velodrome for 5-fluorouracil. When only mutations are used as input, the baseline performs similar to Dakota. One of the key contributions of our work is that it is directly usable in clinical settings after further validation. To the best of our knowledge, we are the first to apply deep co-training task decomposition or Dakota in cancer drug response prediction and showed competitive performance. We also experimented with various input types available in the FDA-approved panels used in clinical settings. We would like to thank AI Singapore for their funding towards this research. Thank you.